Okay, so I realize it's election night, and so other people have certainly justifiably other things on their minds, but we do need to make sure that we are getting our material covered for Chapter 14, our last chapter that is included on our next exam, that exam which occurs online a week from today. So we wanted to do this review session a little bit earlier to make sure that you had time to, number one, take the quiz, which covers our last two chapters, and also that you would have time to get into this information a little bit further and review. We're going to look into the high points of urinary system tonight. Remember that next Tuesday, and I'll send an announcement about it, usual online exam occurring at 8 o'clock. You'll have 90 minutes, somewhere between 8 p.m. and 10 p.m. You need to log in and get that done before the 10 p.m. deadline. It'll have the typical structure where it's mostly multiple choice and then a lab section covering our Physiology labs and then a short answer section. Okay, so let's look at Chapter 14. Now, Chapter 14 combines two different things. It combines the urinary system proper, but then it also combines information on how we regulate or control the water volume or the fluid volume that we have in the body, and also the content of it, the concentration of it. Now, when we talk about concentration or osmolarity, that can be from modifying the water content, but it can also be from modifying the actual amount of the solutes or the electrolytes that we find within it. So we do spend some time towards the end of the chapter going through important electrolytes and how they are controlled, how they're transported, how they're controlled in order to maintain homeostasis. All right, so let's look over some of these major functions. Okay, so urinary system overall is fairly simple in terms of its macroscopic view. We only have a few organs: kidneys, ureters, bladder, and urethra. Now, while on a superficial level it looks pretty simple, we know that as we get further into it, it's more complex. So we're going to zoom in, and we're going to get to this structure here, which is, as this bullet point says, the structural and functional unit of the kidney. So that is the nephron. So the nephron is going to be the powerhouse, the workhorse, if you will, not a mitochondria or anything. It's bigger than that. But it's going to be producing all of the different functions that we ascribe to the urinary system. It's going to be occurring in each of these different nephrons, a million of which make up each kidney. So the nephron is really where we want to focus a lot of our study areas. What's going in the nephron, how does it produce urine, etc. So, focusing on this level is much like when we were learning about either nervous system or muscular system, and we were focusing on like the generation of inaction potential or a sliding filament mechanism or something like that, right? You've got to understand what's going on in the nephron in order to understand how the urinary system works. Now, what we'll find is the nephron is that there's a couple of different types. You can see two different structural types there. They are, it's based on how long their loops are and basically how long they are. So we go into a bit about their anatomy, and then we get into some of their more detailed anatomy. So we start with this thing that we call the renal corpuscle, which consists of the glomerulus, this ball of capillaries, a very specialized ball of capillaries, and then the Bowman's capsule, also known as the glomerular capsule. We look into that because there are three critical processes that occur inside of the nephron, okay? So you need to know all three of these processes. One is going to be filtration, and that's what occurs here in the glomerulus. Two is reabsorption, and three is secretion. Okay? Those are the three critical processes that occur inside of the nephron that allow us to make urine. Okay? So you definitely need to know those three. You definitely be able to describe them. And then when we talk about the different parts of the nephron, the different parts of the nephron in a way are kind of like the digestive system, where you have different digestive organs. Each section has its own particular function or contribution to the system overall. And when we look at the nephron, each section contributes also, but this is on a microscopic scale. Okay? So when we look at the different parts of the nephron, starting out with the renal corpuscle, then going to, if we look at it, let's say we look at it here. Starting out with the renal corpuscle, then going to what's called the proximal convoluted tubule, which is coming right off of it. It's kind of winds back and forth here. Then we go to the nephron loop, or the loop of Henle that we see here. Then it gets all windy and kinky again, which is called the distal convoluted tubule, and that drains into the collecting duct, or the collecting tubule. Okay? Each of those different segments has a specific role. Okay? And so those three functions that we were just talking about are going to occur in particular areas along that nephron. So you need to know where in each of the segments of the nephron, where does reabsorption occur, where does secretion occur, etc. So you need to be able to define, you need to know those, you need to define those, you need to say exactly where they're occurring in the nephron, and then you need to be able to give a couple examples of things that are being filtered, things that are being reabsorbed, things that are being secreted. Okay? These are ions and molecules, because that's what we're trying to do with the urinary system, is control the concentration and the volume of our extracellular fluids. So the first thing we're going to do is we're going to filter things out of the blood, and then we're going to, within the rest of the nephron, spend time and effort kind of tweaking that filtrate, that fluid that has come out. Reabsorption, we're going to pull some stuff back from it, a lot of stuff. Secretion, we're going to push more things into it. So when we say things, you need to know what ions, what molecules, etc. Okay, so that is a major, major part of the urinary system, is knowing the three different processes, knowing the different parts of the nephron, and where those three processes occur and what moves in each section. Another thing that we get into in a topic regarding the nephron is how we control all of those activities, and how the kidney itself and its function is coordinated with other systems. And so the juxtaglomerular apparatus is another important structure that's associated with the nephron. We can see it basically here. That is part of the afferent arteriole and part of the distal convoluted tubule. This whole thing that we see here, this big group of cells, is the juxtaglomerular apparatus. And in essence, it's a checkpoint that allows this part of the nephron to see about the blood that's coming in and compare it to this filtrate after we've run it through the nephron for a while. So it's kind of like a sewage treatment plant where we're checking what's coming in and what's going out. Okay? So you need to know a bit about the juxtaglomerular apparatus and what it does. This figure here shows us our three different processes, shows us the places where they occur. Now this is a very extremely simple representation of the nephron, right? All those little tubular segments are all straightened out and stuff like that. But that at least is getting us to picturing these three different processes and where they occur. So you start with glomerular filtration, because that's where we're filtering, we're pulling things, filtering things out of the blood. So we talked some details about filtration, what causes it. Same thing that we saw movement occurring with other types of capillaries are going to occur here, but instead of exchange, like we saw with other capillary beds, what we're looking for here is we are looking for a net movement that goes out of the glomerular capillary and into the Bowman's capsule or the Bowman's space. Okay? We don't want things going back and forth. We want a net movement out of the cardiovascular system so that we're producing filtration. The main thing that causes that is glomerular capillary blood pressure, right? Hydrostatic pressure or blood pressure that we saw before. So there's so much of a pressure of that, and you see that in the big arrow here. That really outweighs any of the other types of forces that are pushing back the other way. So they cause filtration to occur, and there are different rates of filtration. And one of the ways that we can control the rate of filtration is to change the diameter of the tubes leading into or out of those glomerular capillaries. Okay? So knowing the dynamics of how we can control, like we said, control and coordinate the activities inside of the nephron is pretty important. I mean, it's primary importance for you to know parts of the nephron, what they do, and how we produce urine. And then, you know, these other levels of control and modification are secondary. Not that they're not important, but if you're kind of starting with tiering your studying, you want to get the basics down first. Okay? So filtration, now we're talking about reabsorption. We're talking about different things and how we get them back. And that's another thing, when we're talking about different ions and molecules that we may reabsorb or secrete, you've got to know how those substances are transported, right? So if we're talking about glucose, it's going to be coming across through facilitated diffusion. If you're talking about, if you're talking about sodium, you're talking about a sodium-potassium pump or active transport using ATP. So each ion or molecule has a different way that it's moved across the wall of the tubule, either reabsorbing it from the tubule and putting it back in the blood, or secreting it into the tubule, from the blood into the tube. Now another thing that we're going to be transporting is water. And on the one hand, a lot of people don't understand that. They're like, wow, why would we be getting rid of that much water, right? We want to keep water and stuff like that. Or if we filter things out at the beginning, why do we go through all this effort to get these things back? The filtration, there's so much filtration that goes on. It's necessary that we reclaim about 80% of that filtered water just so we don't end up a dried up husk. And that's called obligatory water reabsorption. Facultative water reabsorption is the rest of that 20% where we can decide, or the body can decide or control, if we want that extra 25% or remaining 20%, or whether we want to let that go. Okay? And that is under the influence of hormones. Hormones will tell the kidney when we want to retain that last 20% and when we want to let it go. And we'll get back to that in a little bit. Tubular secretion is our third major process. You need to know what that is, you need to know where it occurs, and what types of ions or molecules are going to be moved. You know, clearance, micturition, not quite as important. Balance of sodium and water. So this does look into, very important, water balance, right? Input and output should be in balance. So you shouldn't know different ways that we take in fluids, different ways that we lose fluids. But if you look at the overall total for homeostasis, these should be in balance. So we talk about the ways that our kidneys will allow us to reabsorb sodium and reabsorb water. Now those two things are going to be tied together, much like eating a salty bag of potato chips, right, and you feel bloated afterwards. When you move sodium into one place, the water tends to follow due to its osmotic gradient. So not only can we have a direct effect in terms of expending efforts, if you will, in the nephron to allow more or less water reabsorption, but if we move sodium one way or the other, the water will tend to follow. So that can be an additional effect there. So look at the ways that we reabsorb sodium, like we said, which is going to affect water reabsorption. You talk about the specific ways that we reabsorb water and how we allow that to occur. And remember that that is part of the facultative water reabsorption, the optional water reabsorption that is under the control of different hormones like vasopressin, also known as antidiuretic hormone. And then another major topic that you need to understand is how our nephrons and our kidneys can produce different concentrations of urine, right? How it can go from producing, when you're dehydrated, a very low volume concentrated urine to a very high volume dilute urine when you are well hydrated. How does it tweak it? How does it make changes in that way? And we know that 80% of the water reabsorption is obligatory. You have to do it regardless. So it must be options in whether or not we're reabsorbing or letting go that remaining 20%. Okay? So knowing how we can produce concentrated and dilute urine is an important topic in here too. And there's two parts to that. Number one, there's the nephron loop. And number two, there's the last tubular segments, like the collecting duct and even the distal convoluted tubule. Okay? Those two parts of the nephron each have a role in producing concentrated or dilute urine. The first role or the first structure, if you will, that has the role is the nephron. I'm sorry, not the nephron, the loop, the nephron loop. So with the nephron loop, what it does is it produces a concentration gradient. You'll notice here we see it's about 300 milliosmoles out toward the cortex, and it gets up to 1,200 or even 1,400 milliosmoles as we get down deeper into the medulla. It is the nephron loop which produces that concentration gradient, that vertical osmotic gradient there. Okay? Now that's the summary of it. You need to look into the mechanism in terms of how it does it, but that's the role of the nephron loop. It produces this concentration gradient. Now in terms of the last tubular segments, the distal convoluted tubule and the collecting tubule, those tubular segments use the concentration gradient. They use the concentration gradient that is created by the nephron loop. And so, in those last tubular segments, if antidiuretic hormone is secreted, then those little channels, those aquaporin channels, are inserted into the walls of the last tubular segments. And what happens then is that these aquaporins allow the water to be transported across the wall of these tubular segments. It allows us to reabsorb water. Okay? So ultimately what we get if we're pulling all this water out is a very concentrated urine, oftentimes like 1,200 milliosmoles. Okay? So we start out with a filtrate that's like 300 milliosmoles, and we can end up with a urine that is actually 4 times as concentrated. Now if we don't want to reabsorb that water, if we're drinking very well, we have a good input, we want to make sure that we're getting rid of excess fluid. And so we don't want that antidiuretic hormone to come in and tell us to reabsorb anymore. What we want to do is keep those last tubular segments sealed up, if you will, so that any of that filtrate that enters into them just goes straight through. We don't pull any water out, it just goes straight through. It started at 200 milliosmoles or something like that, and by the end of it, it's about the same, right? A very low concentration. So it has to do with those hormones and the insertion of those aquaporin molecules. That's what we see here. Then, that was in terms of controlling water reabsorption and water balance. Then we also said we were going to look at particular ions or electrolytes. So sodium, we need to look into the mechanism of sodium. It starts with what's called the RAS mechanism. You see the renin that is secreted by the kidney there. The angiotensin, and angiotensin 2, which is the first A there, and then aldosterone system. It's a chemical cascade that ultimately causes the release of aldosterone. The aldosterone causes us to reabsorb salt, and we know that when we reabsorb salt, the water will follow, and that will increase blood volume, and that will increase blood pressure. Okay, so you definitely need to know that mechanism as well for sodium reabsorption. Now we saw in a couple of steps with the reabsorption of water from antidiuretic hormone and the reabsorption from sodium from aldosterone, those are going to work to increase blood volume and increase blood pressure. But we also have atrial natriuretic peptide hormone, which has the opposite effects. It actually causes sodium to be excreted so that we get rid of it out of the body, and it decreases blood volume and blood pressure. Okay. One of the ways, like we've been talking about mechanisms for modifying output, you can certainly also modify input. And so the hypothalamus is the main control center for thirst, and so these various inputs to the hypothalamus can help to increase our fluid input if we need it. Now for some of our other ions, knowing the mechanism, knowing where they are reabsorbed or secreted, and if they are reabsorbed and secreted is also important. Potassium is closely tied to the management of sodium because it's the sodium-potassium pump. And so when the sodium-potassium pump becomes activated, we reabsorb sodium and we secrete potassium. So the effect of aldosterone on sodium also has an effect on potassium. Now calcium and phosphate are also important. These are regulated by other hormones, particularly parathyroid hormone. And then we get into other types of ions where we control it, specifically electrolytes like hydrogen ions, right? Because that affects pH. So if we can help to control pH, that's going to be very important for our tissues and our various fluids in the body. So the last major topic of this chapter, like we said, is control of the internal environment and what types of things we're controlling, etc. And pH is certainly an important one there. Okay? So what we need to know is that there are 3 different ways, and we'll see on the slide coming up, that the body can work to control and maintain pH. Okay? One is in the kidney. So what we talked about a little bit previously was the secretion or reabsorption of various things, and hydrogen ions are one of the things that are secreted. And so as you get rid of hydrogen ions, it makes the fluid more and more basic, right, less acidic. And notice that this mechanism that we have here with the carbonic anhydrase, it secretes hydrogen into the tubule, and at the same time, it allows us to reabsorb bicarbonate. That bicarbonate, when it goes back into the interstitial fluid in the blood, can ultimately bind with another free hydrogen and help to buffer pH. Okay? So urinary system is one way that we can control pH. Now, if we don't control pH very well, then there's going to be problems. But here we see renal mechanisms for controlling pH. There are also respiratory mechanisms, and then these things are called chemical buffers. Different chemicals that are in our blood and in our extracellular fluids that can combine with hydrogen ions when we need to and release them when we need to do that. Okay? So those 3 lines of defense kind of work differently in terms of how quickly they work and exactly how they do it, but they all help to maintain pH in the body. Now, if that gets out of whack, then we enter into what is called acidosis if it becomes too acidic, or alkalosis if it becomes too basic. And there's different ways that we can get into those states. If your respiratory system is not working properly, then obviously that's going to lead to respiratory acidosis or alkalosis. If some of these other responses are not working properly, or there's something else going on in tissues, we call it metabolic acidosis or alkalosis. So you need to know what acidosis and alkalosis are. You need to know these 3 different mechanisms: the chemical buffer system, the respiratory buffer system, and the renal or urinary buffer system. So these are different examples of different things that combine hydrogen or let them go. With the respiratory pathway, I mean, we already talked about the urinary pathway, but with the respiratory pathway, let me remind us of that equation that is involved there. So the equation is this: H2O plus CO2 gives us H2CO3, and that breaks down into hydrogen ion plus HCO3, which is bicarbonate ion. Okay? So that's a very critical equation in the respiratory system, because it points out the way that we get carbon dioxide from ourselves and then through this chemical equation turn it into a bicarbonate ion for how it's transported in the blood. But you notice when we do that, it liberates a free hydrogen ion. So the more carbon dioxide that accumulates in the tissues and things like that, the more acidic they become. But if the respiratory system is doing its job, the more the respiratory rate increases and we're exchanging air, then the more carbon dioxide we're going to get rid of, and this equation can go back the other way. So when it goes back the other way, then our tissues are going to become less acidic, because now that hydrogen is going to be bound up instead and turned back into water. Okay? So knowing those 3 lines of defense in terms of pH control is also pretty important. Okay? And this figure right here is a pretty good summary figure, figure 14-5, for the different parts of the nephron and their particular role in our system there and getting it done. Hello, Monique. It's a very interesting thing, as you just showed up right now and you heard me talking and it seemed like I was on the last slide. It reminds me, this is our first instance where we are doing our discussion section, and your state and my state are on different time zones. Oh, because you guys went to daylight savings time, and Arizona doesn't mess around with that stuff. So that was my bad. Being it was the first time, I kind of forgot that that occurs. So our 8 p.m. start time is actually 9 p.m. for you guys, or for me. Yes, so Arizona doesn't do any of the time change. So half the year, California and Arizona are on the same time. The other half of the year, we are 1 hour later because you guys fell back. So when I was here at 8 o'clock, it was 7 o'clock your time, and I waited for about a half hour or so. And then I figured people were watching the election and things like that. And so I just started going and making an archive of our important material related to the urinary system. So what I would say is we already have the kind of focus outline for the urinary chapter and kind of highlighting the important parts and stuff like that and trying to focus your efforts, your study efforts and things like that. But, and that'll be all in the archive. But the question is, do you guys, as you're coming into it, I know it's a little bit earlier, intentionally so, do you guys have questions yourself in terms of urinary system? Things that you were kind of coming to the table with and you wanted to ask. Monique's typing. Christina's thinking. So Monique says she's been looking at the objectives and focusing on those areas. Okay. Christina says, reviewed the lecture notes and topic of juxtaglomerular apparatus found it difficult to fill in those blanks. We can certainly bring that up while we're looking at questions and things. Gotta remember this is the slide there. So we'll leave that up there while we're figuring on questions. So Debbie, as I was just explaining to Christina and Monique, I forgot about the time change, and with Arizona not changing time and not falling back, when nobody was here at a little bit after 8, what I decided to do is actually just go through and do the summary of Chapter 14, even though nobody was here, so that we would make sure that we had an archive of it. So I have gone through the high points of it and worked on focusing on the important aspects of the chapter, so that that'll all be included in the archive that you guys could listen to. But we were waiting to see if you guys had other questions from Chapter 14 also. So Christina's typing again. Monique's typing. Yeah, the study guide, the objectives that I sent, it does cover all 3 of the chapters that we need to cover on the up-and-coming exam. And Monique says, will there be a quiz on urinary before the exam? Yes, indeed. So we already had that first quiz on respiratory system. And so I set up a second quiz that will be half urinary and half immunity, or half lymphatic system. So that one is already up on Desire to Learn. And so that one, I think I have it available through Sunday night. Right now it's totally open. Yeah, so it'll be through Sunday night at midnight. So that way you can make sure that you've taken the quiz and see how it goes and where you need to spend more time. And that way you'll have time to review as well. So Katrina, you were asking about an email about the due date. So that's on the Monday. I don't know if I saw your email. And then what about immune? So all 3 chapters are included on the up-and-coming exam. It's respiratory, immune, which is also lymphatic, and then urinary. The quiz includes urinary and immune. It's half and half. So it's 10 questions, 5 of urinary, 5 of immune, usual 15 minutes. And Christina, I have to take a look in. I believe I got your email, but I'd have to take a look. Go ahead and forward it again. It certainly can't hurt, just in case I overlooked it somehow. Okay, I know I'm seeing it now. So I can take a look at that in terms of the specific questions that it has and get back to you. I knew that I had a few questions come in at that point, and so I sent out that general email in terms of what people can do and how you should kind of focus your efforts to finish up the semester and possibly try to raise your grade. And so for the most part that addressed a lot of people's questions, but I can also look into those couple that you were asking about as well. Other things? Well, since we had that little snafu in terms of the time, and you guys are kind of waiting for the information that's going to come that I already recorded, if you have questions or if you're looking up questions, go ahead and hang out. If you figure you'll just listen to the archive, and then you can email me questions from there, then you can go back to the studying at this point and look for the archive in short order. I'll wait probably another 5 minutes or so just in case other people show up, and then I'll stop the recording and get it posted to the website. Okay? All right, that sounds good.